Well, thanks everyone for sticking around for the last talk of the day. Hopefully it will be exciting enough to keep you awake. I'm from Sunflower. We are a small biotech company in the Boston area, and we are working on perfusion fermentation for eukaryotic microbes, but particularly for Pichia. So that's what I will talk to you guys about today. As I said, Sunflower is a small biotech company in the Boston area, and our goal is to make protein manufacturing more accessible. So a fun fact about sunflowers, they can actually grow in all of the habitable continents of the world, and you don't have to be an expert gardener in order to grow a sunflower. So we think that these same ideas should apply to protein manufacturing. You should be able to do it anywhere, and you shouldn't need to be a bioprocessing expert. So that's a little bit of a background behind our company's name. So the challenge in making bioprocessing more accessible is that conventional bioprocessing looks like this. It's done in a very large-scale plant. It's very capital intensive, labor intensive, and it takes a lot of space. So at Sunflower, our goal is to kind of redefine this paradigm to make biomanufacturing more accessible. So if we think about accessibility, it really comes down to two main things. Something that is accessible needs to be easy to obtain, and it needs to be easy to use. So for something to be easy to obtain, it needs to be relatively low in cost, it needs to be small footprint, and it needs to be deployable and usable in many different environments. For something to be easy to use, it should be simple. These days, probably it should be automated so that it can work very easily without intervention, but also needs to be efficient and flexible enough to make many different things. So in our research, what we found is that there's kind of two key things that really correspond to simplicity and accessibility in biomanufacturing. The first is the host organism, and the second is the manufacturing approach. So big surprise, what I'm going to tell you about the host organism is that Pichia is actually a great option, or eukaryotic microbes in general. So you go to the next slide. I got to shorten this whole section down to one slide for this audience, which I was very excited about, but a couple of reasons that we really like eukaryotic microbes. On the upstream side, Pichia can secrete complex proteins, but it grows really fast, similar to other cells like E. coli. It also has a much smaller genome than, for example, mammalian cells, which makes it easier to work with and sequence if you want to. On the downstream side, the raw secreted proteins can come out at about 60 to 80% purity, which limits your purification needs. There's also 10-fold fewer host cell proteins compared to mammalian cells, so it makes it easier to purify. And there's no contamination with adventitious agents or endotoxins. What this translates to is fast development cycles. So for example, we can go from a protein sequence to a research cell bank in 4 weeks. And for certain protein classes, we can actually go from a protein sequence to a bulk drug substance in as few as 8 weeks. And then, as I'm sure you guys know, there have been multiple approved products already made in Pichia, which makes this a great example. So now I'll tell you a little bit about the manufacturing approach. If you go to the next slide, please. So continuous manufacturing can enable a lot of things, including efficiency. This has been well tested in the mammalian space, so I'm going to use some examples from the CHO space to kind of provide some motivation here. So if you take a look at the bioreactor volume required to make a kilogram of monoclonal antibody in about 1 month using CHO cells, if you do it using a typical fed batch reactor, that's 350 liters. If you do it using a continuous bioreactor, that's only a 50-liter bioreactor that you need. So that's 7 times smaller. Next slide. Continuous manufacturing also enables quality. So what we're looking at here is a principal component analysis. So it's basically looking at a wide dimensional space projected into 2D. And what you can see in the red dots for the fed batch environment is that they're spread out all over the graph, which just means that the cells are experiencing different things throughout the cultivation. If you look at the blue dots representing the continuous process, they're all on top of each other in one spot. showing that the environment that the cells are experiencing throughout the cultivation is much more consistent. This consistency leads to higher cell health and product quality. Next. So what we can see on the top here is a measure of cell viability. For a fed batch process, as you start to build up your waste components, your cell viability will go down, and eventually you have to stop your run because your cells are dying. For the continuous process, you can go much longer and maintain your healthy cells. This often translates into higher product quality. In this example, a product variant was increasing at the end of the fed batch fermentation, but maintained at a low level for continuous fermentation. So overall, this leads to higher quality in both your cells and your product. Continuous manufacturing also enables productivity. So I'm gonna make some people mad here. We don't like to use titer as a measurement. Titer is awesome if you are doing a fed batch or batch bioreactor because it is the amount of protein in your bioreactor. If you're doing a continuous cultivation, you're constantly removing protein from your bioreactor, so you wanna measure that too. So we use what's called space-time yield for a normalized comparison. This is the total amount of protein produced per bioreactor volume per cultivation day. So what you can see here in this space-time yield plot is that because of both your longer cultivation and your healthier cells, you can get higher space-time yields using continuous operation. Let's go to the next slide. So why doesn't everyone already do this? Well, there's a couple of challenges in applying continuous fermentation to microbes in particular. The first one is that the kind of legacy method for doing this is a chemostat. You put your feed in and you pull out a mixture of cells and supernatant. The problem with that is you're constantly pulling out cells, so it limits how many cells you can actually get in your bioreactor. On the kind of mammalian cell side, the way that people have approached this is using a filter-based device that sits outside of the bioreactor. You pull your cells out, separate them from the supernatant, and recycle the cells back into your bioreactor. This doesn't work very well for microbes because they respond to their environment so fast. So if you're pulling them out of the bioreactor, they're losing their oxygen, they're losing their food, and they get unhappy with you. So what we've done at Sunflower is develop what we call an in-vessel cell retention device, which allows us to do perfusion fermentation without removing the cells from the bioreactor. So the way that this works is you are constantly putting your media into the bioreactor. Using our kind of cell retention device, you can pull out your cell-free harvest, which hopefully includes your secreted protein of interest. And then if you want to, you can have a cell bleed. This allows you to kind of keep the cells in that bioreactor environment at all times while continuously giving them food, removing waste, and pulling out your protein of interest. Ultimately, this results in healthier, more consistent cell culture, longer campaigns, very high achievable cell mass, and high levels of protein production. So if you go to the next slide, some of you may have already seen our model in the other room, but we have taken this technology and are commercializing it as the Daisy Petal single-use bioreactor system. This is a benchtop bioreactor system made for perfusion fermentation. So it has a 1 liter working volume, but you can make 15 to 20 liters of supernatant in about a week. We've also really focused on accessibility here. So we have a single-use structured consumable set that's really easy to install. I encourage you to kind of take a look at it and see. You can see how all the tubing lines up with the valves and the pumps so that you can't actually put it in wrong. The system also comes with custom software, which is fully automated to maintain all of your set points, pH, temperature, DO, all of the normal things that you want in a bioreactor, and it's immediately deployable. So it comes with accessories such as an oxygen concentrator, so that all you need are process fluids, power, and cells. Next. We had some early access units that were delivered in the summer of 2022, and we got some great feedback from some graduate students who had never run a bioreactor before, who told us that it was really easy to use and get started with this equipment. So what does the data actually look like coming out of the system? Well, I mentioned that the continuous fermentation can support very high wet cell weights. So in this example here, we achieved over 700 grams per liter wet cell weight. This is shown in the orange line. I compared it to kind of Invitrogen's typical fed batch process in the blue line, and you can see that you can get a little under twofold higher cell mass as compared to that kind of typical fed batch bioreactor. Due to the continuous operation, we maintained it for significantly longer as well. Next. The perfusion fermentation also allows us to sustain high quality protein production. So in this example, we were making a high value cytokine, which is relatively hard to express, GCSF. We operated the bioreactor for 9 days and made over a gram of product in that 1 liter working volume. This translates to a spacetime yield of about 130 mg per liter per day. It's 4 days longer than your conventional batch. And if you kind of put that in perspective, if you were doing a 5-day fed batch campaign, that would translate to about 650 mg per liter titer. And it's nice and clean, as you can see. Next. Another advantage of this continuous operation is that you can actually do multiple conditions within a single fermentation. So if you think about a design of experiments where you might want to do 9 different conditions in your test. Using traditional fed batch, you could do that with 1 bioreactor. It's going to take you over a month. You could do it with 9 bioreactors, but then you have to have 9 bioreactors. With the continuous experiment, you can do it using 1 vessel in only about 1.5 to 2 weeks. Let's go to the next slide. The way that this works is the software allows you to set up recipe steps. So at the beginning of your recipe step, you will kind of set your parameters, give the cells some time to adapt to this new steady state, and then sample from your bioreactor at the end of that time period. You then move on to the next step with your new pH or your new temperature and do the same thing. So using the same bioreactor, same experiment, you have many different conditions tested. So we had an early access customer who demonstrated some of these benefits of perfusion. So they ran their process for about 2 weeks with an average wet cell weight of about 600 grams per liter, making a monoclonal antibody. And through this process, they tested 2 different growth rates and found that they had higher productivities at the earlier growth rate. So in addition to the couple of examples that I've showed here, we've made a wide variety of different proteins using this approach. This includes antibodies and antibody-like proteins, cytokines and hormones, including single-chain insulin, vaccine antigens, and virus-like particles, as well as other enzymes and small binding proteins. We are also working on a larger scale version of our bioreactor called the Dahlia Petal. This system has a 15-liter working volume, but it's basically equivalent to a 200-liter batch reactor because of this continuous operation. And it can sit in less than 20 square meters of space. So we've done just some initial hardware testing on this, making the same cytokine, GCSF, and we showed that in our very short test, the spacetime yield that we got on the Dahlia system was equivalent to the spacetime yield that we got on the same smaller system. So just to summarize some of the benefits of perfusion fermentation using our systems, we have this continuous method, which allows us to be more efficient in both space and time. We have full automation, which allows for hands-free operation of the bioreactor. It has been designed to be as intuitive as possible, so you don't need to be an expert to run it. It's actually more sustainable. Single use doesn't sound sustainable, but because you don't have to do clean in place or other validations, there have been studies that show that single use is actually more sustainable for the environment. It's also flexible. You can make a wide variety of proteins using this approach. It's scalable, and it's ready for use in many different environments. So with that, I'd like to thank the Sunflower team. I brought this picture of us volunteering at an animal shelter because everyone likes to see the puppies, but we have a really great team that's helped put together this whole data set. So, next.
When you run the reactor, is it making decisions on when there's a certain dissolved oxygen set point or things, or is it just following a protocol?
So you set things like the dissolved oxygen set point and how long you want to keep it, and it will automatically adjust the air or oxygen going in to hit that. You basically set everything up as a recipe ahead of time. And you might say, I want to have my media 1 for growth that goes in for 2 days. And then I want to switch to, for example, a methanol-containing media to make more protein. And that's all automated based on the recipe that you input.
Great talk. So for your in-reactor perfusion system, since you have such high cell densities, do you see like fouling or decrease in permeation over time?
It's a great question. We have designed our controls in order to limit any fouling and issues like that. So the cell retention device, along with the proprietary controls, are really what enable the success over long periods of time here. And that's most of what I can say on that topic.
Early on, that your system can produce is sevenfold more efficient compared to, let's say, legacy systems. And then the actual data, you pointed yours was twofold. It yielded twofold higher cell mass compared to the legacy system. Where does the sevenfold come in?
Yeah, so the sevenfold was an example taken from literature where this is done for CHO cells. But it's a great question because the advantages come from 2 places. The advantages come from more cells. The advantages come from more productive cells because they are happier, there are not waste products, and you're actually feeding them a sufficient amount, and from just having a longer runtime. So you don't have to do turnover, you don't have to wait to grow cells again, it just overall increases your facility utilization.